Okay, why don't we go ahead and get started? So thank you all for coming out this morning. It's my wonderful pleasure to introduce Doug and Pam Soltis. These are really, giants in our field and in the field of botany, and they're making faces, but it's true. It's wonderful to have them here. They're going to probably give a really wonderful talk about phylogenetics and the evolution of life in general, but also particularly plants. So Pam got her PhD from the University of Kansas and went on to be assistant professor at Washington State University, where she moved up the ranks to professor. And then moved over to the University of Florida, where she was professor and is now distinguished professor and the founding director of the University of Florida Biodiversity Institute. And Doug got his PhD from the University of Indiana and went on to do postdocs at University of British Columbia and faculty positions at UNC Greensboro, Washington State University, then moved to Florida, distinguished professor at Florida. And they've been really a research team throughout their careers and have won many awards jointly, including the Dahlgren Prize, the Asa Gray Award, and they were both elected to the American Association and Advancement of Science, and also are fellows of the National Academy of Science in the US. So their work has been, they've published over 400 journal articles that have been cited nearly 70,000 times, which is quite amazing. And their research over the years I really respect because it looks at one group of organism plants, and particularly flowering plants, from many different angles, both in terms of what's going on individual species levels, on the genetic and genomic level, on the macroevolutionary level, across the tree of life. So they've looked at particularly phylogenetic relationships in the evolution of flowering plants, the genetic and genomic consequences of polyploidy, and but also conservation genetics, working with museum collections, biodiversity informatics, how we digitize museum specimens, and much more. I could keep going, but I think better for me to just turn it over to Doug and Pam to give you the lecture. So they're going to take turns and jointly give this lecture.
Thanks.
Thank you.
Thank you. Thanks for that really nice introduction. And thanks for the invitation. Can everybody hear me okay? Okay, great. Well, we're really happy to be here. This is a wonderful place that you have here. So I'm going to start off, and then we'll tag off around halfway through, and then Pam will take over, and she'll be happy to answer any of your questions at the end. So our topic today is on the tree of life, building the tree of life, using the tree of life, and then how do we teach people the importance of the tree of life? So let's start out by, what do we mean by the tree of life? Well, we're talking about the totality of biodiversity on our planet, all of the millions of species on this planet, and how they're interconnected through a great family tree of relationships, which we'll call the tree of life. Now, we like to start with this slide. I love this quote: "Behind the man is the tree of life." And I think this sums up really well the concept that throughout human history, the idea, the myth, the tradition of a tree of life seems prevalent in every human culture. It is as old as we are a species. Now, it's different from our modern tree of life view. There's no evolution involved. It was spiritual, it was mystical, it was religious, but it was part of their being. So I think some of these tree of life stories are remarkable, and I'll share just a few of them with you. And some of them, humans are actually thought to be leaves on trees connected to all other species. In other cases, humans are actually descended from a branch of a tree or descended from a tree. Now, here is an artifact. There are quite a few artifacts of the tree of life from the Middle East that go back to 4000 BC. Here's the actual, whoops, I went too past it. Here's the actual artifact here, and then an artist rendition here, just trying to show the connectivity among all species on our planet. But there are also serious religious connotations to this as well. There are also Tree of Life renditions from Asia that go back to 1000 BC, and so those are also remarkable. Now, in the Bible, the Tree of Life is mentioned over 10 times. Some of you may know this, this reading from Genesis. The Tree of Life, however, always gets short shrift to the Tree of the Knowledge of Good and Evil. That's the famous one. But the Tree of Life is there and mentioned over 10 times in the Bible. Now, all of these Tree of Life myths or stories across the globe and through time, they all vary. But there are some common themes, and I like to illustrate some of those common themes by using the Mayan Tree of Life as an example. So with the Mayan, as with many other peoples, they actually believed that there was a spiritual connection to all other species on our planet. Now, importantly, humans were not above all other species on the planet. They were just connected to all other species on our planet, and they envisioned that with the tree. But they actually had a real tree, a physical species that they connected with the Tree of Life. It was the kapok tree, which is in the cotton family. It was one of the largest trees in their forest. And so to them, that was the spiritual connection to the Tree of Life. But you'll see in these Tree of Life, so here's the above ground portion and the branches. There are species here, but there are also various deities and spirits here. Again, remember, this is a spiritual connection to everything on our planet. It also connected them to the underworld. So you'll see similar stories or traditions about the Tree of Life in Northern Europe, for example. Now, another thing about many of these Tree of Life's myths or traditions is that it was taboo to damage or harm the Tree of Life. So the Hopi Indians in the American Southwest, part of their Tree of Life story is that there was an earlier civilization that exploited or overexploited the Tree of Life, and it ultimately led to their demise. Something words to live by today. Now, how important was this Tree of Life to other humans? Well, they actually wore it as part of their clothing. Here we see embroidery. These are Tree of Life symbols that are from the Huron tribe. This was from the North American Northeast. This was a very prominent tribe, part of the Cherokee Federation, Iroquois Federation in the northeastern North America. But these are Tree of Life depictions. And some of these symbols carry on today. So the Turkic people, which are common people, a very common tribe throughout Central Asia, and their descendants are in places like Turkey today. We see their Tree of Life symbol still surviving in the flag of Chavezia, which is part of the Russian Federation. And this coin from Turkey, you can see the resemblance of that to one of the early artifacts that I showed from the Middle East. So these symbols still survive today. But of course, our modern concept of the Tree of Life dates to the time of Darwin. And many of you may know this quote from The Origin of Species. And it's so beautiful, I'm going to go ahead and read it. "And so by generation I believe it has been with the great Tree of Life, which fills with its dead and broken branches the crust of the earth and covers the surface with its ever-branching and beautiful ramifications." Wow, that's gorgeous. You can't tweet things like that. Really. And notice that he covers the extinct, the dead and broken branches, as well as living species as well. Now, this concept of the Tree of Life was so important to Darwin that the only illustration that he insisted on in The Origin of Species is not a map of the Galapagos Islands, it's a tree. And this is that figure from The Origin of Species. Now, we have ancestors, descendants, some go extinct, and then existing species here at the present. Now, this stimulated other scientists of his time, such as the famous German biologist Haeckel, to begin to draw or depict the Tree of Life, as you see here, which is, Haeckel, as many of you may know, is an amazing artist. And so some of his renditions of how he thought life was related on Earth are striking. They're beautiful. You know, they would make great posters on your wall. And here is one of them here. But again, remember, this is his view, his gut feeling based on years of experience of how life on Earth was related. And it just shows the major groups of of life. Now, amazingly, that tradition of using your lifetime of wisdom accrued and hand drawing a tree carried through for at least another century. Here is a tree of Arthur Cronquist. He was a famous American botanist, showing how he thought the major groups of flowering plants were related to each other. And you can see he actually named them, he used a series of bubbles, so this has become known as Cronquist bubbles. And then within each one of these bubbles, he would show a branching diagram of how he thought the major groups in each bubble were related. So that was well over 100 years after Darwin. And it really wasn't until very recently, 2015, that we were finally able to construct the first rough draft tree of life for all 2.3 million species that have been named so far on our planet. And here it is. So the origin of life would be here. Here's an ancestor, and these are all descendants. Actual living species, all of them are out here on the periphery of this circle. I'll explain the colors later. We'll come back to that. But notice that arrow. That's you. That shows our place, Homo sapiens, on the tree of life. Notice we don't get a gold star. Our branch isn't any bigger than any other species. I like these big trees because they're humbling. They actually show our place in the world of species that surround us. Okay, so if Darwin were in the audience today, he said, what took you so long? Why did it take over 160 years to build what is really just a first rough draft? I'll keep stressing that word. It's not final, rough draft, tree of life. And the answer to that has several components. One is that unlike other disciplines, physics, chemistry, astronomy, this is really a very recent field. We had no theory for building trees until the 1960s, and this German entomologist, Willy Hennig, really provided the first underpinning of theory for building trees. By that, I mean actually using data and building a tree in some way that somebody else could use the data and it could be reproducible, not just drawing how you think everything is related by hand, which had been the tradition, as I said, for well over 150 years before that. So that introduced tree thinking, which really has been pretty common now for, when you think about it, just a few decades in the field of biology. So it's been a remarkable gig, if you want to say that. It's been a remarkable journey for the past 3 or 4 decades to see tree thinking take over and have such an impact on so much of biology. So a tree is a phylogeny, and I know sometimes, we always like to confuse everybody when we draw trees because we do it in a bunch of different ways, but you can have the ancestor down here, and then here's a descent. And these are ancestors, all ancestors that are extinct, inferred ancestors, and then living species are here. Or here's a tree of lice. Yeah, lice also have a tree of relationships. These are the living species, the ancestors here. This node's an ancestor. You can draw them as sort of sticks, or as I said, these circle tree with the ancestor here. And those are always sort of striking and beautiful things. But the other reason that it took so long to build the first rough draft tree of life is building big trees is really, really, really hard, rivaling the most difficult problems in physics, math, and astronomy. And why do I say that? Well, when we build trees, we now use computers, and they sort through the data, and they try to come up with the best solution to the data, okay? So if you have 3 species, there's only one way you can put them into a tree of relationships. That's pretty easy. With 5, there are 15 ways. With 10 species, it's over 2 million. I think you can see what's happening here. With 20 species, the number of solutions to that problem, that is the way you can put those into trees of relationships, is now approaching 1 mole. Oh, get it? 6.02 times 10 to the 23rd. That's one of those things, that probably all of us still remember from chemistry. It's embedded in our brains. And I knew I would finally get to use it someday. And so here's how we did it. And then when you get to 200 species, now the estimate is the solutions to that problem, the number of possible ways you can put those into trees is exceeding the number of atoms in the universe. So you can see these are really tough problems. And so it's no wonder that in the 1970s and 1980s, I know that's before some of you were born, there were actually papers published in prominent journals, you think of it, Nature, things like that said it was really going to be impossible to build a comprehensive tree of relationships for all life on Earth in some sort of single analysis. So building the tree of life was considered some sort of grand challenge in biology, or a moonshot for the study of biodiversity science. But ultimately, we did it. And by we, I mean a large community of scientists, not just a few people. It took evolutionary biologists, experts on discrete groups of organisms, computer scientists, et cetera, to combine and come together to build this first rough draft tree of life, kind of reflecting modern science. So much of it is now team-oriented. And that's really what this took. It was really, the timing was right. It was a perfect storm. Algorithm development, computer power, which we all know so well. And then, of course, the DNA sequence revolution was really key, because the key element in building these trees of relationships, DNA sequence data, that is really one of the most important tools in building these trees. So with that in mind, I'm not going to say much about graph theory. If you want to know more, I'm sure there's maybe a mathematician in the audience that can help. But we all use graph theory. And graph theory was really fundamental as a breakthrough in building these really, really big trees. Just as you're connected to everybody through Facebook, you can connect all of the species on Earth through graph theory. And so that was really an important development. So how big is a tree of life? I think people have trouble grasping how big a tree 2.3 million species is. Well, if I print it on a piece of paper, 12-point font in a linear fashion, it would take the Empire State Building, 4 sides of the Empire State Building, 14 Empire State Buildings, all 4 sides, to display the tree of life in a linear tree printed on standard notebook paper at 12 font, okay? So it's enormous. That's why we use these circle trees. We actually have a tree of about 6,000 species printed, and it would stretch out of this room, down the hallway, and probably out the building. But the point here is that we have so far to go. What we're showing you really is just a beginning. It is just a starting point. This quote from E.O. Wilson, I think, points out how many species we have yet to discover. It is entirely possible that specialists have discovered only 20% or fewer of Earth's biodiversity at the species level. And of course, we're in a race to find as many of the surviving species as possible before they vanish and are overlooked and lost forever. But that's only one part of the problem. The other part of the problem is shown here. Let's come back to this tree that I showed you earlier, and I said I would come back to the colors. So let's come back to the colors. Here is our tree of relationships for 2.3 million species. I said DNA was a fundamental tool in building trees. Where we have lots of DNA data, it's shown in red. Where we have no DNA data, it's shown in blue. And you can see this tree is mostly blue. In fact, only 17% of named species have a snippet of DNA that's useful for building a tree of relationships. So you can see how far we have to go, even with the life we've already named and described on this planet, to get it into a meaningful tree of relationships. Now, some scientists estimate there may be 10 million species on this planet, maybe more. That is really biased towards complex cells, organisms like us, animals, plants, fungi, eukaryotes that have complex cells. What it doesn't take into account is the bacterial world. And when you consider the bacterial world, there may be over 100 million species or more on this planet. So I know some of you may know the song by Madonna, "We're living in a material world." She almost got it, right? We're living in a bacterial world. And in our tree of life, we only added named species. Bacteria usually aren't named, and so they're not in the tree of life, but not very many are in that tree of life. So that's a real underrepresented group. Now, of course, everybody thinks, well, there's so many undescribed, unnamed species in the tropics and other remote areas, and of course that's correct. But another point that I think is really important to make is that there are a lot of species right under our noses in very well-studied areas, whether it be Asia, Europe, North America, that have gone undetected until maybe now we're just figuring out that there are a lot of what we call cryptic species. So I know we have butterfly experts out here, insect experts, and so they'll know the answer perhaps to this. But there are actually 4 species of butterfly on this slide, okay? Each row is a different species. It's just male, upper, lower, female, upper, lower. Those are 4 species. They were distinguished by DNA. They certainly can tell each other apart, even though by eye you can't tell them apart. But cryptic species are common in all lineages. Here are 2 frogs that look pretty similar, but they have distinct calls, so they do not interbreed. And in the plant world, cryptic species abound. Here are two flowering plant species that if you were to look at them here and here, the leaf, wow, these look really the same. But they have different chromosome numbers, they have different geographic ranges, one down here in black, one up here in white. They don't interbreed, there are no hybrids. So it was really only very recently that these were named as two distinct species. So there are hundreds, thousands of species like this in the plant world that have never been named in well-studied areas like California and other places in the world. Well, let's move on a little bit and talk about the value of the tree of life. So I think many of you may know this famous quote by Dobzhansky: "Nothing in biology makes sense except in light of evolution," which is a great statement. We probably all use it a lot. The corollary to that today is that everything makes a lot more sense in light of a tree of relationships. What do I mean by that? Well, I think we all get this. We know that our own family tree matters, right? We know that relationships matter, so that if this is your family tree and you are here, you realize that if a close relative has a disease that's genetically inherited, say this person here with the lightning bolt, and if you have a strong probability of inheriting that gene, right? So these are predictive. Trees, family trees are predictive. And in the same way, our tree of relationships, or any tree of relationships, of species relationships, is also predictive. It has that power. Relationships matter. So we can use the tree of life for many, many things, whether it be drug discovery, crop improvement, disease, the study of disease conservation, climate change, ecology, biodiversity, and more. And so now what we're going to do is give you a few examples of some of these ways that the tree of life can be used. So let's consider first medicines. Most of our medicines come from plants, of course. But the point is that there are many, many medicines yet to be discovered. And how do we discover those? Well, the traditional way was to just sort of go through one by one and chemically assay a plant and look for active compounds. That is tough when you think that there may be 500,000 green plants alone. So a better way to do that, perhaps, is to use the tree of life. Here we see a tree of relationships for the flowering plants. It's a simple version. There may be 350,000 species of flowering plants alone. But if you want to look for the next generation of medicines for heart ailments, nervous disorders, these two places are these are where you want to look. Those are the spots to look. Why? Because there already have been a number of active compounds from those lineages, and we know that there are big groups, and by folk medicine and traditional medicines, we know that there are many other plants in those groups that also have active compounds and can be useful. So in fact, some pharmaceutical companies are already doing this. They're tapping into the tree of life as a way to explore and come up with the next generation of medicines. Crop improvement. The tree of life is so essential in crop improvement. This statement by Miller of the USDA: "If no germplasm from wild relatives had been used, there would probably not be a viable sugarcane industry in any place in the world." That's pretty profound, and that really sums up the situation for most of our crops. Where do we obtain new genes for disease resistance, for increased yield, for improved water use and efficiency? Where do we get them? From wild relatives. How do you determine the wild relatives? You have to build a phylogeny, or a tree of relationships. So here's an example from our lab. A student worked on squash. I think many of you may know some of these. We've got pumpkin here, we've got crookneck, we've got acorn squash. So these are all really mesic plants. They require quite a bit of water to grow. So how do you build a better squash? How do you use those? How do you come up with a squash that may be more efficient in its water use? Well, what our student Heather Rose Cates did is that she built a tree of relationships and actually found out that the closest relative of those squash on the previous slide that requires so much water, there are two species in the arid Southwest. That's a real cactus back there. You can get an idea. This is a pretty dry area, and she's collecting germplasm now for the USDA so that this can be used in breeding programs. So this is how the tree of life can be useful. Now, trees of relationships are essential in the study of disease. I don't know how many of you remember the SARS epidemic, 2002, 2003, started in China. Over 700 people died. There was this fear. It was spread to 37 countries. So there was this fear that this was going to be this amazing, huge pandemic. But where did SARS come from? Well, it was not originally in humans. The original host was another organism. And using a tree of relationships, it was found that czars, so these are different strains of czars. They're shown in a simplified tree. Here's an ancestor, descendant, ancestors, and descendant. You can see that the closest relative of czars and humans is this civet, which is a small mammal, a species from Asia, and then maybe also bats. But this honed in. This is where we would look for where that jump occurred from one host to another to humans. Now, this is how other diseases are going to be monitored. This is the way that you would study and be ready for the next pandemic. Also, trees of relationships are used every year to try to predict the next flu vaccine, which if you didn't get, now you know, or if you did get, when you do get a flu vaccine, a phylogeny was used to try to infer how to make the best vaccine from one year to the next. So let's move on now and start to cover topics of conservation. So when you go to the restaurant and you think you're ordering tuna or mackerel or some other fish, how do you know after it's in sushi or topped up or something that it's actually what you ordered? Well, you don't. And this is where a tree of relationships can be very, very handy. So, a lot of these fish species, DNA is available. They've been put in a tree of relationships. All you have to do is scrape some fish off your plate, get a DNA sample, run down, sequence it, and insert it in a tree, okay? Well, actually, people did that at a number of restaurants in the US in big cities, Pittsburgh, Chicago, San Francisco. And guess what they found out? About 47% of the species on the menu were misidentified, okay? So you're not always getting what you think you're getting. So there's real conservation implications here. You can similarly use a tree of relationships to see if in logging in areas of Southeast Asia, for example, if endangered species are being illegally harvested, you can do the same thing. So it's also used for detective work there. All right, another conservation example. This little bird is called the white-winged warbler, which is really hard to say 10 times fast, white-winged warbler. You can try this afterwards. But using DNA in the tree of life, it was determined that the white-winged warbler, guess what, is really not a warbler. It's actually, it just looks like a warbler. It's a distinct lineage of birds. Adding to its conservation concerns, it only occurs on one island, the island of Hispaniola. So all of a sudden, this little bird's conservation status just shot way up the charts. Distinct lineage only on one island. Whoa, whatever it is, if it's not a warbler, we still have to, this is really an important species to conserve. Okay, now this may be one of my toughest slides to explain, so hang in there, bear with me. We can also use trees of relationships to really kind of infer what lineages of organisms may be particularly susceptible to a changing climate, which is, of course, really important right now. So this is a tree of relationships. It's on its side, so the species are over here, but we've removed all of those because it really doesn't even matter what the species are. The ancestors over here. But if you want to know, there are plants, there are trees in this group, there are shrubs, there are little herbaceous things like this that are some of the most cold-tolerant plants in the world. You can see that's really ice and snow in the background. So it's a highly diverse group of plants. So what we did is that we took information that we could obtain for the mean annual temperature at which these species occurred, okay, and we substituted that on the branches of the tree, and then we used, then we tried to infer the ancestral state of this group of organisms. And you can see the colors. If it's red, it's hot adaptive. If it's blue, it's cold adapted. So our hunch is this group of plants probably started out in the tropics, probably trees, probably in between adapted, right? And then you can see this is 100 million years of evolution. It spurred off some hot adapted lineages and these blue lineages that are cold adapted. What you need to see is how canalizing those events are. When you switch to cold adapted, whoop, you don't switch out. When hot adapted, you don't switch out. So these lineages have been in that state sometimes 15, 20 million years. They are not going to suddenly switch to warm adapted in the next 20 years. It's very, very unlikely. Now, similarly, we can do this for other groups of organisms. The Okinawa lab, Evan, we talked to you yesterday, you're doing similar things for ants. So we can actually use sort of a global perspective global, in several ways, other lineages of life, and also the globe as a whole, to predict what lineages of life might be most susceptible to a change in climate, whether it's increased temperature or decreasing moisture. Okay, and so at that point, I'm going to turn things over to Pam, who will take us the rest of the way.
Tag. Okay, thanks. So for the next few minutes, we're going to explore some of the responses of plants to climate change and how the tree of life can aid us in conservation decisions. Now, we're going to be using examples from Florida because Florida has an amazing diversity of plant species. Now, you might think of Florida as home to Mickey and Minnie and their friends and a large number of vacation spots, but it's also home to more than 4,000 species of vascular plants. And many of these species occur in very specialized habitats and are very susceptible to climate change. And the work that I'm going to be describing is the result of a number of collaborations, but most prominently with our colleague Rob Gralnick, who works right down the hall from us, and Julie Allen, a former postdoc, and also a former grad student at the University of Florida. She now has her own lab at the University of Nevada, Reno. Now, for many pressing societal problems, we look to new data, new methodologies, and new sources of information. But for some of our problems, such as things related to biodiversity, we actually have a huge legacy of information that's stored in our natural history collections. And in fact, there are somewhere between 3 and 4 billion specimens estimated worldwide in natural history collections. Now, we may tend to think of these as specimens that are just dusty old things that are locked away in cabinets. And until very recently, that was probably a good assessment. And in fact, most specimens have been locked away in these cabinets, unavailable for use by specialists, scientists, or use for education or by members of the public. But recent efforts in the US, in Europe, in Asia, in Australia, basically worldwide, in digitization are now leading to exposure of these data in ways that can make this information really useful for all sorts of biodiversity-related problems, including conservation. So how does this work? Well, here's a plant specimen from a herbarium, and this pressed plant specimen has a label. And this label, if it's a really good label, will tell you all sorts of things. Of course, it will have the scientific name, but it will also have the date of collection, the collector's name, the location. Any recent collections will probably have GPS coordinates. If it's an older collection, hopefully there's a textual description that will allow us to actually use software to pinpoint where exactly, or exactly with a range of uncertainty, that specimen might have been collected. It might tell us about associated species, and there might be additional notes. Now, that's the ideal world, and many labels, luckily, do sort of follow this ideal. So this information from millions of specimens is now being compiled in data aggregators around the world. And in the US, iDigBio is the National Center for Digitized Biodiversity Collections, and we're working with other organizations such as GBIF, the Global Biodiversity Information Facility, and also other large aggregators like the Atlas of Living Australia to merge the information across these resources so that there will be multiple ways to get at the biodiversity data that the world's natural history collections house. Let's see. So what does this mean for conservation of Florida plants? Well, we can actually take the location information from these herbarium labels and use those to extract environmental data for those particular localities where the species were collected. And we can extract temperature, precipitation, soil, geochemistry, et cetera, from global databases. And then we can use software to model the range of each species. And so for Florida plants, we've done this for about 1,500 plant species out of the total of the 4,200 that are estimated. And this is work that was also done by a former postdoc, Charlotte Germain Aubry. And just to give you a few examples of what this looks like, we've got two plant species. This first one, the flat spike sedge, not quite as hard to say as the white-winged warbler, but it's getting there. This is a grass relative. If we build a model for this species, you can see that by the warm colors that its most suitable habitat is in southeastern Florida, not a particularly good location, given that this is sort of the center of Miami, and this is a large urbanized area. In contrast, this is the scrub plum. Its most suitable habitat is from the high elevation areas of Florida. This is the Lake Wales Ridge, which sort of forms a backbone along this area. And these are what the models look like for the present. We can also use climate predictions for the future to estimate what the species distributions would be under models of climate change. And so for the flat spike sedge, we see that in fact that bit of warm color that is present here has actually disappeared. And there's really not any suitable habitat predicted for this species in 2050 as the conditions get hotter and drier. Now, in contrast, some species that are already adapted to hot, dry conditions, such as the Lake Wales Ridge scrub area here, will actually have the potential to expand their distributions. So whether or not the species could actually expand to this great range is another question, but at least there will be suitable habitat over a much larger area predicted in 2050. Now, we made these same sorts of models for, again, about 1,500 of the plant species, those for which there were enough specimen records to satisfy statistical properties of the modeling. And we get a map like this, where green represents high species diversity and the tan represents areas of low species diversity. We can then also do this for the future and then compare current diversity with changes in that diversity in the future. So this shows actually a shift in diversity. So green areas will be areas that will be suitable for an increasing number of species. So there's the possibility of movement into those areas. So we see this area, which is the Everglades area of South Florida, being a potential recipient of species, and other areas such as in the northern part of the state and then moving northward throughout the eastern U.S. also as suitable habitat. Whereas some of the areas that are currently high diversity areas are going to become less suitable for species. So it looks like there will be in the future the possibility of some species moving northward and other species moving southward, at least in terms of where their suitable habitat is. So how do we use this sort of information to design conservation strategies? Well, we have a number of biodiversity hotspots globally. There are 36 global biodiversity hotspots. Within the US, there are several as well, most prominently in California and also in Florida and the southeastern coastal plain. There's also high species diversity in the Appalachian Mountains in the eastern US. So how do we use information from biodiversity hotspots and other sources to develop conservation strategies? Now, typically, conservation decisions are based on the total number of species in an area, or perhaps on so-called indicator species. But what if we could use the power of the tree of life to contribute to conservation decisions? Now, when we examine biodiversity hotspots, such as those in California or Florida, et cetera, we can also start to look at how much of the tree of life is actually present in a specific area. And we can use something called phylogenetic diversity. And phylogenetic diversity is simply a measure of how much of the tree of life is present in a geographic area. Now, how does this work? Well, let's say we have one area that has 8 species. And these 8 species are all oaks. Now, if you're an oak taxonomist or maybe even a squirrel that gathers acorns, these 8 species may seem very different to you. But if you're not an oak specialist and you don't really care very much about oaks, this might seem like 8 species that are very similar to each other. Now, if in contrast we have a second area with 8 species, maybe a few of which are also oaks, but we have other things like a water lily or a snapdragon or a bellflower as well, then this area would capture much more of the overall tree of life than this area with just its narrow little circumscription of these 8 species of oaks. So the second area would have higher phylogenetic diversity than this area. And some people have argued that we might want to be using phylogenetic diversity as a metric for determining which areas merit conservation more than other areas. Trust me, it's coming. For some reason, it's a slow thing to emerge. So let's say we have an area here, like this little dot in the panhandle of Florida. We have a species list for that area, and we can map the species from that area onto the phylogeny for the plants of Florida. And everywhere where you see red, that marks a branch or a part of the tree where those species occur. And we can then compute phylogenetic diversity for that area. So what do we need to do a phylogenetic diversity assessment? We need a phylogeny. So here's our tree for the plants of Florida. And we need locality data, which we can get from herbarium specimens as recorded in iDigBio, for example. And then we can compute the phylogenetic diversity for each of those little red dots, actually every little grid cell that we could divide an area into, and we can determine a map such as this. So this has areas of high phylogenetic diversity, the darker areas here, 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 and so on, and areas of low phylogenetic diversity, such as this part of the Everglades, this part of northern Florida, and so on. So we can then take these patterns of phylogenetic diversity and consider them in the context of other environmental parameters. And we can also compare them with aspects, for example, of human population growth. So here we can see areas that have low phylogenetic diversity and low population density. These blue areas, such as this one here, which is again the Everglades. We're not saying that the Everglades should not be conserved. There are many other important reasons for conservation of the Everglades ecosystem, but simply on the basis of phylogenetic diversity and low population density, this would not necessarily be a prime area for conservation. We also have areas such as those in green that have high phylogenetic diversity but low population density, and that actually covers a good part of the central part of Florida. And these might be prime areas for conservation because there's high phylogenetic diversity in terms of the plant growth and not a lot of conflict from human populations. Of course, that's changing rapidly as 1,000 people per day move to the state of Florida. Most critical, however, are these areas in this brownish red color. They are areas with high phylogenetic diversity, but also high population density. And we see that in the Miami area, in the Tampa Bay area, in the Orlando area. And these are areas where there certainly is need for conservation, but because of the human population density and patterns of urbanization, it might be very difficult to actually use this information to set aside conservation areas. Well, how well do our conservation areas actually capture areas of phylogenetic diversity? So here is our phylogenetic diversity map I showed you before, and here is a map showing the conservation areas in green, including offshore areas here in the Gulf of Mexico and this very large region offshore in the Atlantic. Now, these three conservation areas actually have low phylogenetic diversity for plants when we compare them with this map here. And so although there may be other reasons, important reasons for conserving these areas, the criterion of phylogenetic diversity would not suggest that those particular areas be conserved. Now, as we think about what areas to preserve in the future, we should look toward areas that do have high phylogenetic diversity and yet are not preserved. So these include mostly areas here shown in turquoise, where we know we have high areas of high phylogenetic diversity, but these areas are not yet under conservation. And so I think as we move forward, there are good opportunities to use the power of the tree of life in our conservation work. Now, this same approach can actually be applied to much larger geographic areas than simply this peninsula, the state of Florida in the US. And here, along with some Chinese collaborators, we examined patterns of phylogenetic diversity across China. And we found that areas of the highest phylogenetic diversity are located here in blue and pink in South China. And yet most of the areas of conservation are located here, much to the west, in areas that are made up not of these complex subtropical forest ecosystems, but instead are relatively species-poor areas of grasslands. Now, species are disappearing rapidly despite our best efforts. And as Daniel Kozlowski wrote, "What your parents can hardly remember, you will not miss. What you now take for granted or what is slowly disappearing, your children, not having known, cannot lament." And we certainly see this as truth in our own families. We think about stories that our grandparents may have told to our parents about particular species. Well, if those species are no longer there, our parents can't share that information with us. Likewise, we can't pass that on to our children. And so in the course of a few very short generations, we can lose knowledge and appreciation for biodiversity very quickly. Now, in fact, extinction rates are about 1,000 times what would be considered normal or background extinction, with about 100 to 200 species estimated as going extinct each day. And just for comparison, I've added in here the maybe 6th mass extinction. These are the 5 mass extinctions that have been recorded in geological history, and the spike shows the number of species rapidly being lost just over the very recent history. Now, in fact, our current geological time slice has even been given its own name due to the human impacts on the environment, and this, of course, is the Anthropocene. So what do we do? Species are being extirpated at alarming rates, and we're continually trying to save those species that remain. E.O. Wilson suggests that perhaps we should set aside half of the Earth's surface in order to establish conservation areas. And perhaps this is a valid approach. But any sort of large-scale action requires education. As H.G. Wells wrote, "Civilization is a race between education and catastrophe." Or, if you're from our era, you might remember this chorus from a very famous song by Crosby, Stills, Nash & Young, "Teach your children well and feed them on your dreams." So we decided that science alone is not going to convince people that we need to take major action, for example, setting aside half of the Earth for conservation. And so we decided to explore some collaborations with art and artists to try to convey our messages about the importance of the tree of life. And science can influence people's minds, but art, in all of its forms, can touch people's hearts. So our first foray into collaboration between science and art was with the Harn Museum of Art at the University of Florida campus. And with the Harn, we established a small exhibit called the Tree of Life, and we used a number of the biodiversity-themed collections within the Harn to expose concepts of the Tree of Life. And there were opportunities for people to move about the museum, addressing questions about the Tree of Life using not just the artworks that were hung in this particular exhibit area, but other pieces of art that were spread throughout the museum. So this was a really engaging opportunity. It led to some additional collaborations that then helped bring scientists and artists together across the campus at the University of Florida. But it was very small scale. More recently, we've been developing an artistic work called One Tree, One Planet, and this is a project that is ongoing. Now, One Tree, One Planet is an artistic projection developed by Parisian artist Nazia Mastawi. And she's an artist, architect, and environmental activist. And here you can see this projection art on the back side of the Harn Museum of Art. This is about a 3-story building. This is a projection of the Tree of Life that was then a public event. Now, this work is a series of 15 vignettes on biodiversity. Each vignette is a story of a different species, and each vignette is set to music based on the DNA sequences of that species. The goal of One Tree, One Planet is to illustrate the connections among species and the connections of humans to the Tree of Life. And we're hoping that through various showings of this work that people will also become more interconnected with each other. So we'd like to share one of these vignettes with you now. So as I mentioned, there's a series of 15 of these stories about different species, and they run sequentially, but with the aid of some human interveners. So this shows a group of folks who were attending the first showing of One Tree, One Planet at the University of Florida, and it shows them engaging with the projection by taking pictures of the projection itself. But what you can also just barely see is the fact that there's a podium here. And that podium contains an iPad that allows for an individual person to interact with the Tree of Life projection, one person at a time. And of course, people stood in line, dozens long, to be able to participate. So the next step is actually to develop a smartphone app. So not just a single person can interact with the projection and with the audience, but many people can interact and connect. And this will be launched in just a couple of months, and we'll be having the app launch at the University of Florida. But then the first real show will, involving the app will be at the Atlanta Botanic Garden just a few days later. Now, we're hoping that there will also be showings in New York and St. Louis. We're working with botanical gardens in both of those areas to set up showings in 2020. And how about here? We'd be ecstatic to be able to share some of this work with this wonderful institute here to have public outreach and scientific input about the Tree of Life. So our second major project involving the combination of science and art is a 15-minute video that we developed with colleagues at the University of Florida in the Digital Worlds Institute. They're graphic developers, graphic designers, animators, musicians, just an amazing group of very creative people. And this video called Tree Tender is actually available at treetender.org. You can watch the movie by going to that website. But it's also been translated now in the sense of having subtitles into Korean. We had the launch of the Korean version last week. There's a Chinese version. Spanish is finished now, and Portuguese is just about finished. So we're extremely excited to work with collaborators to develop these alternative ways to reach the public in different parts of the world. Now, we showed this also first at the Florida Museum of Natural History. Over the course of 2 nights, we had the One Tree, One Planet and the Tree Tender movie, and we had over 1,500 people present. And again, this is out of a relatively small university town in Florida. And then also these materials are available online, and we know that many, many more people have been observing them there. Now, rather than show this entire 15-minute movie, we are going to show you the trailer from it.
Good morning. You must be the new curator. I am a catalog assistant and virtual interface node. Welcome to the Tree of Life. You can call me Calvin.
What is the benefit of having the tree?
Salix babylonica is one benefit of the Tree of Life. This tree has medicinal properties of great value to humans. Scientists were able to derive new medicines from Salix babylonica, such as acetylsalicylic acid.
In other words, aspirin.
Correct. You are experiencing chronophylogenetic travel as we move between organisms. The Tree of Life illustrates the relationship between all species on Earth.
You're saying this tree shows every single thing in existence?
Correct.
And again, the entire movie is available at treetender.org. Now, finally, we'd like to close with a quotation from that great biodiversity scientist, Albert Einstein. Look deep into nature, and then you will understand everything better. Now, the work that we've described today is the product of many collaborations. Doug described some of these when he was speaking. I've mentioned several of our other collaborators. But these all represent many, many large projects, the products of collaborative science. And the art and science work in particular, I'd like to point out as being, having been supported by the Florida Museum, various offices at the University of Florida, and Digital Worlds. These are the main collaborators for the art and science collaborations. And then the science work that we've described has been supported in large part by the U.S. National Science Foundation. And with that, I'd like to thank you very much for your attention, and I'm sure Doug will answer any of your questions.